interferon. Look at the whiteboard. This is I have written here IFN. Interferon is abbreviated as IFN. Interferon. You can write interferon or IFN. This interferon. Let us see the definition of interferon at first. Interferon is antiviral substance. This interferon was reported for the first time by Sir Isaac uh, Isaac in 1957. The interferon was coined by Isaac. Interferon with it interferes with the multiplication of virus in the host cell. Now let us discuss point out some other important features. This interferon is secreted by the infected host cell. Its receptor, you will be surprised. There is a very important point which you must know, that the receptor for this interferon is located in the cell membrane of all the animal cells. This interferon interferes with the further multiplication of virus in the adjoining in the infected and adjoining cells. This interferon induces synthesis of ABP. How does this interferon act? Interferon does not act directly. Rather, this interferon acts by producing another very important protein, and that protein is known as the ABP. I have written here ABP, antiviral protein. Antiviral protein. You should not confuse here. This interferon is also proteinaceous in nature, but as far the mechanism of the functioning of this interferon is concerned, the interferon itself is the proteinaceous in nature, having high molecular weight. But this interferon, how does this interferon act? This interferon acts by inducing the synthesis of another protein, and that protein is known as ABP, which is produced in the adjoining cell. Now let us see the different features of this interferon. Let us see. I have written here the features of the interferon. Interferons are protein at first. You must know the biochemical nature of this interferon. Interferons are all proteinaceous in nature. They have a high molecular weight. The molecular weight of the interferon varies from 8,000 to 1 lakh Dalton. This interferon are produced against the viral infection. This interferon remains stable for the several hours at the room temperature. Interferon is a stable protein, and it can be stable for the several hours at the room temperature. They are host specific. It is not a virus specific. You know that the interferon produced interferons are host specific. They are not the virus specific. And a particular, what does it mean? It means that the interferon produced in the host cell is different from the host cell, different from the interferon produced in another cell. So therefore, you can say that the interferons are the host specific, and it is not a virus specific. This is another important character. Now let us see the next character. These interferons are more stable than the antibodies. You know that antibodies are produced against the antigen, but the interferons are more stable. As far the stability of this interferon is concerned, it is more stable protein than you all know that antibodies are also proteinaceous in nature, and interferons are also proteinaceous. But which protein is more stable? Interferons are more stable than the antibodies. They have the interferon have complementary binding site located on the adjacent cell. I have told you that the cell membrane of all the living cells have the receptor for binding with this interferon, and they have the complementary binding site located at the adjacent cell. Now look at the next character. Interferon induces the cellular genes to produce the ABP, which blocks the transcription of viral. Let me tell you to make you know that how does this interferon acts as antiviral substance, and what is the mechanism? This interferon induces cellular genes. You know, just I have told you that the genes for encoding interferons are located not on the viral genome. Rather, they are located on the genes for encoding the interferon are located on the host cell genome, and interferon induces, therefore it is mentioned here that interferon induces cellular genes to produce ABP. antiviral protein which blocks the transcription of viral mRNA. When, as you know, that when a virus infects any host cell, the viral mRNA gets transcribed on the host cell ribosome to produce the proteins and the viral genome also gets multiplied there, and fresh viruses are produced. So what does this interferon do? This interferon prevents the binding of viral mRNA with the ribosome to prevent the to prevent the to prevent the transcription of the viral genome. And next, this interferon prevents the binding of ribosome with the viral mRNA, but does not affect the binding of the ribosome with the host cell genome. What does it mean? It means that this interferon has capacity to block the binding of mRNA with the ribosome, viral mRNA with the ribosome. It has nothing to do with it does not affect the binding of host cell mRNA with the host cell ribosome. So it has the capacity to block only the binding of viral mRNA with the ribosome. It does not prevent the binding of the host cell mRNA with the ribosome. So this is very important feature. Next, binding of interferon with the receptor sites also affect the change in the cell membrane of the dividing cell. When this interferon binds with its receptor located on the cell membrane, it brings about the changes in the conformation of the cell membrane. So this is another very important feature of the interferon. Let us see the next feature. This interferon prevents the polysome formation. What is polysome? Hello, Paja. What is polysome? Paja Anjum. Paja.
Yes, sir.
What is polysome? Polysome. Do you know what is your concept regarding the polysome? Where this polysome is produced and what does this term signify?
Sir,
ठीक है, राइबोसोम से हाँ बताओ क्या होता है थोड़ा सा. You are just approaching toward the answer. ठीक बता रही हो, you are trying your best. ठीक है, तुम बताओ राइबोसोम से ही रिलेटेड है. यस बोलो.
यस सर, ग्रुप ऑफ राइबोसोम मतलब बहुत सारे राइबोसोम मिलकर पॉलीसोम बनाते हैं.
तो तुम यानी यू मस्ट, ठीक है, लेकिन थोड़ा अच्छा से इसका यू मस्ट राइटली डिफाइन इट that the binding of a number of ribosomes with the same mRNA. When the same mRNA, when a number more than one ribosomes get bound with the single mRNA, that such this structure constitute the polysome. Generally it happens in case of bacterial cell. Bacteria में generally होता है, देखोगे कि एक ही mRNA से बहुत सारे राइबोसोम कभी-कभी बाइंड कर जाते हैं. ठीक है, तो उसको पॉलीजोम कहा जाता है. तो ये दिस इंटरफेरॉन आल्सो प्रिवेंट, दिस इज वन ऑफ द फीचर, दिस इंटरफेरॉन प्रिवेंट्स द पॉलीजोम फॉर्मेशन. पॉलीजोम बनने से रोकता है. दिस इंटरफेरॉन प्रिवेंट द पॉलीजोम फॉर्मेशन इन द एडजेसेंट सेल. Next character, next feature of this interferon is this interferon sets off the activities of cellular enzyme meant for the replication of the viral DNA या viral genome. When the virus infect the host cell, ठीक है, the viral genome replicates itself inside the host cell. So then what is the role of this interferon? This interferon prevents, this interferon sets off, switches off, this interferon switches off the activities of the cellular enzyme meant for the proliferation और replication of the viral genome. जैसे इंटरफेरॉन क्या करेगा कि इंटरफेरॉन वायरस के जो एंजाइम जो रिक्वायर्ड है वायरस के डीएनए को या वायरस के जीनोम को रेप्लिकेशन के लिए होस्ट सेल में, उस एंजाइम्स को ये ब्लॉक कर देता है, उस एंजाइम की एक्टिविटी को सेट ऑफ कर दिया. ठीक है, तो दिस इज अनदर वेरी इंपॉर्टेंट फीचर ऑफ द इंटरफेरॉन. इंटरफेरॉन्स आर, अब देखो, नाउ लेट अस सी द इंटरफेरॉन्स आर ऑफ वेरियस टाइप्स एंड ऑल दीज इंटरफेरॉन्स हैव बीन ब्रॉडली क्लासिफाइड इनटू two major groups, type 1 interferon and type 2 interferon. Type 1 interferon include two different types of interferon, alpha interferon and beta interferon, and type 2 interferon include the gamma interferon. And I will discuss the differences between the alpha and beta interferon and gamma interferon in the next lecture. So these interferons are classified into various types depending upon their किस basis पर, depending upon their chemical nature and antigenic property. दो basis, there are two bases for the classification of interferon. The first basis is that their chemical nature, and the second basis is their antigenic properties. So these are the some very important. There are so many important, so many features of the interferon, but these are the some of the very important features of the interferon. So in your examination, you will be asked to write, and you will be a full question may be asked. What are interferons? Describe their types and the production and their application. So or agar interferon, what are interferon? Discuss their types and mention their application. Okay, so application interferons are very important chemical. These are being used, they are being produced on the commercial basis, pharmaceutical basis for the treatment of viral diseases. And the name of the this is the type 1, just I am mentioning alpha, beta and gamma interferon, but you will not get this product in the market by the name alpha, beta and gamma interferon. Rather, the trade names are different. Beta serons, beta interferon is the trade name by the name by which this product is available in the pharmaceutical companies or market. This is the beta seron is the chemical name, trade name for the beta interferon. There are so many. Similarly, we will be discussing all the different types of interferons and their trade name and their production and their application in the treatment of viral diseases. And the very important, one of the most important disease which is treated by use of this interferon is the multiple sclerosis, hepatitis. For the treatment of hepatitis, this interferon is being used. And not only this, even for the treatment of common cold and influenza, viral influenza, this interferon is used. So the interferons are very important. What are interferons? Interferons are how will you define the term interferon? Interferons are the antiviral substances, and they are produced by the infected host cell, and in the adjoining cell. And this interferon was for the first time discovered by Alfred A. Isaac in 1957, and the term IFN was coined by himself. And we have also discussed the various features and some of the important features. I again want to repeat you that interferons are antiviral substances. They are proteinaceous in nature. They have high molecular weight. Their molecular weight varies from 8,000 to 1 lakh Dalton. The interferon have their receptors located in the cell membrane of all the cells. The very important character is that interferons are host specific. They are not virus specific. This is another very important character. It is not a host virus specific. And again, there is still very important character that the genes for encoding these interferons are not located on the viral genome. Rather, the gene for encoding this interferon are located on the host cell chromosome. Host cell chromosome पर जीन जिस host cell पर अटैक करता है, उसी host cell के अंदर we all of us have the genes located on our chromosome for encoding this interferon, but it is not, these genes are not expressed. When these genes are expressed, when the virus infect the host cell, the host cell has the genes for encoding the interferon genes get activated, and they encode this protein which is known as the interferon. This interferon is host specific. Just I have mentioned, interferon induces the cellular genes to produce ABP. And another very important character is that the interferon acts through production of another protein which is known as the ABP, antiviral protein. Okay? It prevents the binding of ribosome with the viral mRNA. Okay? It also affects the cell membrane by bringing about some conformational change. Binding of interferon with the receptor sites also affect the changes in the cell membrane of the adjacent